and welcome to day 5 of our R programming session. Today we are going to investigate a couple different things within R, but really focus around interpreting t-test comparison differences. So we're going to start with defining a function. I wanted to note that I've incorporated the same function 2 different ways here at the top of the code, and I wanted to try to explain why they differ. The short version is that we want to be able to compute a large number of t-tests all at once. So if I state how different my set of numbers are, I want the software to simulate a whole bunch of data that allow us to evaluate, can we detect a difference if the standard deviation difference is x? Okay? So at the top, we see that we have these 2 functions defined. But I think to get to a place where this is a little more comprehensible, I might actually start with some visualizations of the normal distribution. So let's start by using base graphics to make a quick visualization of the normal distribution. So I'm going to grab this section of code out of the file. This input script should be visible to you if you're joining us from the web in a URL that appears in the description below. So I'm going to grab that section of text, I'm going to pop over to R, hello R, and paste it in. And right away we have our image. So I think if you've ever had a statistics class, this is probably at least somewhat familiar. The 2 characteristics that we use to define to parameterize a normal distribution is the mean value, which is the center, and the standard deviation, which describes whether it's fat or skinny. In this case, I've drawn a purple line at its mean value, which is right here. This is a mean value of 0. And the standard deviation in this case is 1. I've shown a single standard deviation by these 2 red lines, and then we have 2 standard deviations out shown by the blue lines. So if I were to try to interpret these data by saying, how commonly would I find a result within a set distance, I might start with kind of the most basic of questions. So what fraction of the data in this normal distribution fall within 1 standard deviation? Does anyone know that value? It's usually around 68%. So if you were to integrate all the space from here to here within a standard deviation, you would account for about 68% of that distribution's values. So if you have a number that's normally distributed, 68% of the time it's going to fall within 1 standard deviation. That's more or less the definition. If we were to allow ourselves up to 2 standard deviations in either direction, we'd be coming out to the blue lines. What fraction of this of this normal curve shows up within 2 standard deviations of the mean? 95, exactly, yeah. So about 95% of the times, 19 out of 20 times, you should have, if you randomly choose a number from a distribution, it should fall between those blue lines 2 standard deviations away. And if you allowed yourself up to 3 standard deviations away, you'd be up to about 99%. So we have a pretty rapid fall off then. We account for about 2/3 of the data by allowing for up to 1 standard deviation. About 95% of the data, 19 out of 20 of the data, if we step out to the blue line, and if we allow for another line at 3, we would expect to have 99% of the values. So that's very helpful. When we're doing a t-test, we're comparing the data from potentially 2 different distributions. The null hypothesis says that they should all be coming from 1 distribution. And the p-value is just a rating of whether or not that null hypothesis holds up under scrutiny, basically. So let's return to our code and try this next example. In this case, we're not going to show just 1 distribution. We're going to show 2 different distributions. All right, let me see. Did I have a... Oh, I want to walk through the code just a little bit that we used on that. You've seen me use a variety of different functions to produce a range of numbers. For example, runif gave us a random sampling from the uniform distribution. Our norm gives us a a sampling drawn from the normal distribution, but I don't think I've used the sequence function yet, have I? I don't think I have. Okay, so let us look at, let's look at this one. So sequence, and I want numbers in this case ranging from -4 to 4, and I'm going to specify a length of 10. 10 is a lot easier to show on screen, I hope you don't mind. So the sequence function is going to return a series of numbers that start at its minimum value, that runs to its maximum value, and that includes this many items. In this case, I gave it a total range of 8, from -4 to +4, and I told it to give me 10 numbers. So we see that they're not distributed so very neatly. Let's instead make this number 8. I think that's going to be a little cleaner. Oh, wait, hold on a second. I botched a thing. Ha, 9. Sorry, I wasn't thinking. Fence post problem is what we call that. Okay, so if I tell it to give me 9 values ranging from -4 to +4, it's going to return me the set of integers that are evenly spaced from -4 to +4. And when I told it 8, I forgot that I needed to count 0 as a stopping point in there, basically. So if I give it -4 to 4, 9 numbers, it's going to give me back 9 values that are evenly spaced across there. Before, I had used rnorm as a way to grab back a series of numbers that came from a normal distribution. When I used dnorm, however, I was using the same distribution but in a different way. So let's run the help on dnorm to remind ourselves of what that means. All right. I've now drawn back I've got help now to show me that I can get the density or the distribution function, the quantile function, or a random generation. So because the rnorm, dnorm, qnorm, and pnorm functions are all so similar, they list them all together. So you can see that dnorm under value gives the density. So in this case, we're working with an ideal normalized, an ideal normal distribution. The software knows that it should be shaped perfectly like this. We're not sampling it, and I'll try to explain why that's different. If I do a histogram of rnorm 100, say, it's kind of normal, but not perfectly normal, is it? So if I wanted to clean this up, I might instead increase up to 1,000 data points. So I'm doing a histogram of a random sampling of 1,000 numbers drawn from a normal distribution. Now that looks pretty clean, doesn't it? Now we're getting to a nice smooth curve. If I keep going, this time to 10,000 data points, well, it still looks a little unusual, I think, but as you can see, as we increase the number of values that we sample, the closer it gets to this ideal shape for the normal distribution. So sometimes you don't want to randomly sample what this distribution looks like, but actually get the ideal distribution out. And that's what the dnorm function does. Okay, so let's return to our sequence of values here. I can return the dnorm values from this sequence of values, and you can see that it's reporting back the ideal density of the normal curve for each of these points, assuming that I have a mean value of 0 and a standard deviation of 1. So when I plotted it, I was using just these first few lines here. So I'm going to show just that first section of the code. So this is the part that draws our normal distribution. It starts by creating a series of x values from -4 to +4, evenly spaced. It's going to give us 1,000 values. It continues by saying, how much density should I have in an ideal normal distribution for each of these x coordinates? And then I plot it using the x values and the dnorm values on the y-axis. But here I'm specifying that I want to draw it as a line graph specifically. See, I had some options when I did that plot. if I left that last bit off, if I just plotted x versus hx, I would get something that looks like this. You see, each of those dots, each of the 1,000 dots, are all overlaid on each other because they intersect with the next one. So doing it as a dot plot doesn't really make sense. This really should be shown as a line graph. So by adding in that type equals L, I'm specifying that it should be drawn as a line connecting each of those 1,000 points together. It looks like a curve because it's just connecting points that are very close together with very fine resolution. But there were a couple other things that we added to that. So if this part plots the normal distribution, we can also add lines on top of that graph. And that's what the AB line function does. You'll remember that I started with a mean value where I drew at a crossing point of 0 on the x-axis. And that AB line is producing this vertical line cutting through at 0. I then had 2 more lines that I drew on at 1 and -1, specifying a color of red for those. And finally, I drew on a couple blue lines. So I'm sorry if I just rocketed past that. I thought it was important to come back and explain how we got that plot to begin with. Okay. Now for this next part, we are going to put 2 normal curves on top of each other. So for this, we're going to start our sequence of x values at -4. This time we're going to go up to 5 on the x-axis, and we're still going to put together 1,000 points. Now we're going to compute 2 different densities. We're going to subtract 1 from the mean in one case, but in the other case we're not. So the first distribution has a mean of 0, just as we saw before. The second one's going to have a mean of 1 instead. We'll then plot one on top of the other. So let's run this section of code. I'm going to close that image, get us back to our text window. Okay. Oh, and I think I forgot a thing. Sorry. Oof. All right. There we go. All right, so what I've told the software is that I want to look at 2 different normal distributions drawn on top of each other. One has a standard deviation that's a mean that's 1 higher than the other. So the red curve represents the normal distribution with a mean of 1. The black distribution here represents the normal with a mean of 0 instead. Now, why have I made this visualization? Because we're going to be talking about t-test. So I want you to imagine that we have some sort of difference in a set of numbers that we're attempting to t-test. If the 2 cohorts that you're going to compare have the same mean, then the null hypothesis is true. Do people see that? Okay, if there's no difference between means and the standard deviations are the same, then the null hypothesis is true, and we shouldn't get a significant p-value, except that we do. We're going to talk about that in a minute, so I'm going to try to explain why. In this case, I'm showing kind of a very minimal difference. If you've done a mock treatment on some cells and a treatment with a very, very small concentration of some drug on those cells, you might expect there to be a very slight difference in whatever you're measuring between these 2 cell populations. So in this model that we're showing on screen, we're making a little model of what that looks like. The means of the standard deviations of these 2 are identical. They're both 1. But the means are very slightly different, a mean difference of 1. So if you were to collect a whole bunch of values from red and a whole bunch of values from black, you might be able to tell the difference. You might be able to tell the difference. But it's going to be difficult. So the bigger the difference that you're looking for, the bigger the difference in the means, the better your chance of detecting that difference. But there are other things that come into this. Now, I'm not going to lurch into a long lecture on power here, but this is the basis of a power discussion, essentially. If you need to know how many replicates do you need to do in order to tell a difference apart, you first need to have some idea of how big a difference you're looking for. In this case, a mean difference of 1 with a standard deviation of 1, you can see that there's not a whole lot of difference here. If you have 1,000 values from black and 1,000 values from red, you can probably tell the difference. If you have 3 values from red and 3 values from black, you're probably in a world of hurt. So remember that finding a difference means that there is a mean difference to start with, and that the standard deviation is small enough that you get a clear separation of the A's from the B's, or the blacks from the red in this case. Everyone sees that. All right, so I'm going to continue on to our next example. When we were looking at that code, we were looking at a mean difference of 1 that was determined by this x minus 1 business. Now we're going to look at a mean difference of 2. Otherwise, our code stays the same. Oh, you'll notice that I've used the lines function in order to add that second plot. So this plot function creates the initial image, and then the lines function is adding the second curve to it. So I should have explained that along the way. Okay, so now we're going to run this block of code, and this is going to give us a slightly bigger difference. This, we remember, is a mean difference of 1. Now I'm going to run that code. Now we have a mean difference of 2. All right, again, if you have 1,000 values from red and 1,000 values from from black, you have a very good shot of spotting even a very small mean difference. Now with a mean difference of 2, do you think that we might be able to get away with a smaller number of replicates than 1,000? I think so. You could probably get away with 10 replicates and do a pretty good job. But how many do you really need to be able to tell apart a standard deviation, a mean difference that's as big as a standard deviation or as big as 2 standard deviations? That's really the question I want to go after. If we run a whole bunch of t-tests at a given mean difference, do we detect them to be different or not? All right, so I'm going to return this back to mostly text mode. That's most of what we're doing graphically today. Now, we're going to start again with something that I want you to know about, but which you might find very difficult to work with. And this is the apply function. You know, of course, that if you have a whole bunch of values in A and a whole bunch of values in B, you can add them together by just saying C equals A plus B. That's all very sensible. It is a little more difficult, though, to reconcile yourself to something like the apply function. The apply function is intended for you to do things like apply a function for all rows of a table. So I've written the same function 2 different ways here. I'm going to start with the easy way, if you don't mind. So I'm going to create a function, just as we did Pythagoras a couple days ago. This one's going to be called TtestPercent. So TtestPercent is going to give us 100 p-values and ask how many of them are below 0.05. That's its function. So at first, I'm going to have to provide 2 parameters to this function to run. One is the n value. This is the number of replicates. Now, since many of you are in grad school or are now doing postdocs and so on, I'm sure that at some point you've been involved in an experiment design discussion with a professor. Do professors tell you to perform 1,000 replicates of red and 1,000 replicates of black? No. Ah! Great. Okay, so we've already had a couple reasons. You don't do that. So Mariska has lent us PI experience. That's very good, principal investigator experience. She says we don't have the money. All right, that's good. So if you're doing a proteomics experiment, are you going to send 1,000 samples over to CPGR for analysis? No, because that's expensive. It's way expensive. At $100 or 1,500 rand a piece, you're going to spend a lot of money doing that. But what's the other thing? Why doesn't a grad student look forward to a n equals 100 versus n equals 100 experiment? You want to go home tonight. You want to go home to your weekend and have a restful time uninterrupted by experiments. Time is expensive, right? So when you're trying to figure out the n for an experiment, you need to have something that's adequate to power that experiment, something that's going to be enough replicates that you would be able to adequately reject the null hypothesis if in fact they are different. Why do we do 3 replicates? I actually get a really common answer on this one that actually is wrong. That you have to do 3 of A and 3 of B in order to do a t-test. It's not actually true. You can measure variance with less than 3 points. It's just that it's an appallingly bad estimate. But why do we settle on 3? To be on the safer side. Well, it's certainly safer than 2, I'll say that. But is 3 enough? No? Maybe no? Surely no? 3 is enough. Going more than 3 is fine, but 3 is enough. On what basis do you say 3 is enough? A better chance of good result than 2 replicates. It is certainly better than 2. But I'm going to contest that 3 is not often enough. 3 is very often not enough. As you just saw in the visualizations that we were producing, I think I don't have that open now. I'm going to reopen it. I can just paste it. Boom. All right. Let us imagine that I have sampled 3 values from black and 3 values from red. Am I going to detect that these are different? The answer is not, of course I'm not. Of course I am. This is a 2 standard deviation difference. It's a pretty big, what we call effect size. Unless you know the magnitude of the difference between one cohort and the other in advance, it's not really possible to discern how many replicates you need. Unless you know how much variance is present in the biological system, you don't really know that 3 is enough. If you don't know how much technical variation you have, 3 may not be enough. And so anytime we do a power calculation, we have to think about things like, with what probability am I willing to accept that a difference does exist, but we didn't detect it, versus claiming that one exists when one doesn't? There are lots of things you need to know before deciding on an experimental design. And so very frequently, Mariska's answer drives things. We do 3 replicates because it's less expensive than doing 5 replicates. But very frequently that means that our experiments are underpowered, that we have so few replicates that we can no longer find a difference, even when one exists. All right. You do 5, that's good. I argue when I was working in CPUT, a proteomics organization associated with the National Cancer Institute, a funded cooperative agreement, a statistician named Dean Billheimer, very sharp guy, once told us 5 is the new 3. So if I could help you with that, I would say, if you remember 5 is the new 3, you will often provide yourself quite a lot more power than what you had with just 3 replicates. Okay, because sometimes the null hypothesis is true. Sometimes you just didn't have enough replicates to detect that difference. 5 is a lot better than 3. So I'm hoping that our little experiment today will convince you of that. All right, now let us start with our function. I'm going to go ahead and copy this to make sure all of this just runs properly. Okay, here we go. So this is the body of the function. I'm going to just copy that in here, and it gives me back nothing, which is to say it has now accepted a new function called TtestPercent that was defined by the function. Always makes me laugh to say that. Okay. So in this, we're going to pass it two different values. One is the n, which is to say how many replicates do we intend to do from each cohort. One is the difference. This is the actual difference in mean values between the black and the red distribution. We're now going to generate 3 columns. You can see number of columns is equal to n, so 3 columns of 100 values each. Their mean value for the A values is going to be 10, and then the mean value for the B's will be 10 plus whatever difference was provided. But we're going to end up with 3 columns of A's and 3 columns of B's for each of 100 rows. We're then going to initialize a variable. P-values is going to be 100 long, but it's going to start out being not applicable, not applicable, not applicable, not applicable. We're going to fill it in as we go, in essence. We're now going to loop from the first row to the 100th row. Is this making sense, or am I just running past? Sort of.
If you don't have that length of that 100, if you don't have that.
Okay, yes. So how do we know that we have 100 values to work with? Okay, up here, you can see that I asked for a bunch of random numbers, and I asked for n times 100 values. So, and I specified that they were going to be in n columns. So in this case, we're telling the function that if we were doing an n of 3, we're going to do 300 values and dump them into 3 columns. So there's necessarily going to be 100 there. I didn't set up this code to let me say, well, now I want 1,000 p-values or 100 p-values or whatever. It's always set hard to 100. Yeah. So in this case, we're simulating a bunch of data, and we're going to ask, do we find a difference or not? We're going to do it with 100 different tests, all meeting that same criterion. These means are this far apart, we have this many n's. All right, so we start by setting the p-values to nothing. They're just set to not applicable, not to 0. We're then going to run from the first row to the 100th, and in each case we're going to set that particular value of p-values to whatever came back from the t-test of comparing the A's to the B's. Now, this is a relatively bad way to do this in R, but it is on the other hand a little easier to understand than the canonical R way to do it. So we'll look at that in just a minute. But once we've run through all 100 rows, we're now going to say, we're going to call the significant p-values anything below 0.05. It's a standard approach, right? And we're simply going to return how many of these p-values were significant from the function. Okay, so let's check to see whether this was sensible by running our new function TtestPercent. Your number will probably be different. I'm going to run this a few times. I'm just hitting the up arrow and hitting enter each time. So what we're asking, remember there are default values in place for this code, which specifies that if they don't tell me otherwise, I'm going to use 3 replicates and the mean difference is 0. All right, so if you were to compare a t-test of 3 values and 3 values all drawn from the same distribution, should those values reveal a difference or not? So I don't think you followed my main point here. I'm specifying a mean difference of 0, meaning the black values and the red values are drawn from exactly the same distribution. So if t-test tells us there's a difference, if it tells us that the null hypothesis looks very unlikely in this case, that actually represents kind of an error. That these distributions are the same, so the null hypothesis is accurate in assessing this. We are generating 100 p-values for this case, and what it's telling us is that 3 of the 100 times it saw a significant p-value. 3 of 100 times, 5 of 100 times, 2 of 100 times, 2 of 100 times, etc. Does that mean the t-test function in R is broken? It's not broken. This is how t-test is supposed to work. That under random conditions where there's no difference between the distributions, you can still get a p-value that's less than 0.05. You should get a p-value less than 0.05 5% of the time, which means that in this case, the p-value should be less than 0.05 5 times every 100 t-tests you run. That's disturbing and frustrating for people, but this is the nature of the beast. Okay, so we've now put in our function, and we've got it working, and it's reporting how many significant p-values come out. Now let's look at this code written a different way. Okay. So again, we're going to create a function called TtestPercent. We're going to create a matrix of 100 values times 3 replicates, or n replicates. We're going to generate another set. This is all exactly the same between the two. Again, we're going to bind together the random A's and B's to create randoms. Oh, sorry, this is actually different in this code. So in this case, we in the case we did before, we set up a different table of random A's and a different table of random B's. In the case up top, though, we stuck them together. You see this cbind function? This is sticking the two matrices together side by side. Okay, so we then, in one step, bypassed all of that for loop. Rather than go from row 1 to row 2 to row 3 doing the same functions over and over again, we use something called the apply function. And in the apply function, we're telling it that the first n columns are going to be our A's, and the columns after those will be our B's, and we're going to run the t-test on it, keeping the p-value. Apply is designed to let you run a function in parallel across an entire matrix, in this case. I find the code is a lot harder to understand when I use it, so frequently when I'm doing the coding, I'm going to use a for loop because I understand that. But apply is something that experienced R programmers would use all the time. There are even things like row apply and stuff like that. But in both cases, we would call out the significance by generating a subset of anything that had a p-value less than 0.05. Okay, questions before we move away from the function stuff? Okay, so the rest of this is going to be about statistics, not about R coding. But I wanted you to have at least one example where you've seen the apply function unrolled in this way. A for loop is one way to bypass having to use the apply notation. It's probably very slightly faster to use the apply notation than to do it as a for loop. I find it difficult. Yes?
With the previous example that you were showing us, what you wanted us to take away from this is that it doesn't matter the number of times you run a test to always be significant.
Okay.
A little bit.
All right, what I'm saying is that you can, when there's no real difference, t-test can sometimes tell you that there's a real difference. Because the null hypothesis, sometimes the data don't look like the null hypothesis, even when the distributions they're drawn from are not different. So these are what some people would consider false positives. We got a p-value of less than 0.05, but the A's and B's were not actually different from each other. It's just that because we sampled only 3 replicates out, under certain circumstances, they looked like a difference existed even when one wasn't there. All right, so if I go to the script here, we can ask, do we get the same number of false positives every time? So here we're going to run 100 t-tests with each line of code. So in the first case, I had one claim that the null hypothesis didn't hold up out of my 100 times. Here I had 3 cases out of my 100 where the t-test gave me a significant p-value, even though there was no difference. 1, 4, 4, 1, 1, 0. Here's a case. 7 times out of 100 times that I ran this, the p-value for the t-test was below 0.05, even though there was no difference. So sometimes t-test can lead you to believe that there's a difference, even though there isn't. All right, now let's try for a very small difference. You remember the first visualization, we had 2 normal curves side by side that had just a difference of 1 standard deviation between the 2. We're going to try going after that with a t-test involving 3 replicates. Okay. So now the difference is real, right? Getting a significant p-value is what you expect because there really is a difference. But did we always get a difference when we had a 1 standard deviation difference between the 2 sets of numbers? It's more than before, isn't it? Right, so we have some number of these that we're able to generate some number of less than 0.05 p-values even when there's no difference. When we have a slight difference, 1 standard deviation apart, we see that somewhere on the order of 12% of the time, we're able to detect that difference in the t-test. But is it always the case that we detect the difference even though it's real? It's not even half the time. So if you have a difference as small as 1 standard deviation and you go after it with 3 replicates, your ability to detect it in t-test is pretty bad. Pretty bad. Okay, so let's continue on to the next case. Oh, I'm going to just keep going with this. So we started with n equals 3, difference is non-existent. Here we have the number of replicates is 3, but the difference is 1 standard deviation. I'm going to just add this in here. TtestPercent n equals 3, difference equals 2. Okay, so the difference between our 2 distributions is changing. We've gone from 0 to 1 standard deviation to 2 standard deviations. And now I'm going to run a bunch of 2 standard deviations, 3 replicate tests. Have we even made it to a 50% chance of detecting that difference? Okay, so if you're using 3 replicates, your ability to detect a real difference of 2 standard deviations is pretty low, not even a coin flip. So it may be that even though everyone knows you do 3 replicates, that's not enough to dig out a true difference, even of 2 standard deviations between the samples. That could mean that your t-testing in experimentation is only going to detect really, really big differences, and you could be blind to real differences that exist in the data. All right, let's work through the same, this time setting n to 5. Okay, so I'm going to start with TtestPercent, n equals 5, difference equals 0. Okay, so I'm going to start with the assertion that there's no real difference between the distributions. Does t-test lead me to believe that there is one? How do we feel about that? Are we glad that we're getting p-values below 0.05? We're not glad, because in this case, the true difference was 0. So it's still possible with more replicates that the p-values are going to be less than 0.05. The distribution of the p-values that come back from t-test is uniform from 0 to 1 when there's no difference. When there's no difference to be found, you should find that the p-values range from 0 to 1 uniformly, which means however many replicates you have. If you have 1,000 replicates, we can work that problem. If you have 1,000 replicates, you're still going to have a fair number. 1 in 20 of your p-values are still going to be less than 0.05 when no difference exists. A lot of people don't get that. And they think that I've got a value of below 0.05, I'm going to publish. But it is plausible that you're fooling yourself, because it is possible to get, it's likely to get 1 in 20 times that you get a less than 0.05 value even with a bazillion replicates. All right. Yes.
The percentages that we are seeing here, it's representative of a p-value. So
Yes, we're asking how many of 100 p-values are less than 0.05.
And say in the first case it's 6%.
Six of 100, yes. Literally 6 of 100, it's 6%, yes.
And then you're saying that is not, it's not a good sign because we're not supposed to detect anything.
In this case, we told it that we wanted there to be 0 difference in the means of the two distributions. They're the same distribution, in fact. They both have standard deviations of 1 in this case. So the reds and blacks completely overlay each other, and yet when we ask t-test, are they different, we get a p-value below 0.05, 6 out of 100 times. That is the expected behavior of t-test. All right, so having done a bunch of 5-replicate t-tests with no difference, let's now try 5 replicates with a mean difference of 1 standard deviation. How are we doing? Was that better or worse than what we had when we had 3 replicates with 1 standard deviation? I'm going to zoom back up a little bit. Okay, so a typical number for this is a little better than 20, which is to say that about, let's say, a fifth to a quarter of the time, we detect the difference with 5 replicates. Now let's look down here at n equals 3, difference equals 1. Was it worth it doing 5 replicates rather than 3 in this case? So a typical number for 5 replicates was a little better than 20. So about 1/4 or 1/5 of the time, we were able to detect a difference that existed. In this case, where we did just 3 replicates, we were barely above 10% average. So our ability to detect a real difference that exists went way up. Wasn't great, but it was way up when we did 5 replicates rather than 3. And now, I'm going to skip right up to this last example. We're going to look at 5 replicates, but this time with a standard deviation difference, a mean difference of 2. I will get my story straight here. Were 5 replicates able pretty consistently to detect differences when they existed that were a mean difference of 2 standard deviations? Yes. Is that ideal? Okay, 3 out of 4 times we were able to detect a difference that existed with 5 replicates and a standard deviation difference, a mean difference of 2 standard deviations, Okay, so Tendeka has expressed her satisfaction with this. She said, that's great. But I want you to think about this a different way. Let's just say that our ability to detect a difference correctly when we have a mean difference of 2 standard deviations is about 3 times out of 4. What if I pick 4 grad students at this table and I say, one of you failed to detect a difference that was real. The other 3 of you did. Now 1 student is not graduating and 3 are. You see why this matters? If you design an experiment with too little power, even if the difference exists, you may not detect it. And on the other side of this, even if you perform lots of experiments, you may claim a difference when none exists. If you do a bunch of multiple tests, if you test a great many hypotheses and seize on the one that happens to give you a p-value below 0.05, and ignore the fact that you've done a lot of hypothesis testing, don't do multiple testing correction, you could claim a difference when none exists. Science is fighting against this all the time. Lots of scientists are using underpowered experiments where they don't do enough replicates. Thus, they miss differences that are real. And many people are not using multiple testing correction, so they claim that differences exist when they don't. You know something today that lots of graduated PhDs in the field of biomedicine do not know. And I hope that when you're having an experimental design discussion with a professor, and he or she says, well, this is expensive, we're only going to do 3 replicates, that you say, look, it really, really matters that we detect this difference if it exists. Can we find some way to test a smaller number of hypotheses, but test them more rigorously? Let's do 5 replicates here. It could make the difference in whether that project succeeds or fails. And it could be your success or failure that you're talking about. So,
What if you're buying, maybe you're buying just a sample?
yep,
Just one sample for 50,000 rands.
yeah.
And what to say, you want to run 5 replicates.
What if it's really expensive?
Yeah, really expensive.
Yeah,
And this was what you're saying, well, I can't afford this with my grant or whatever.
Right.
So what are you doing?
what are you going to do? Yeah, that's an everlasting question. Doing an underpowered experiment invests money, but it doesn't get an answer. Or it gets a wrong answer. No difference exists. Right? So spending the money and not coming away with valuable information is worse than not doing. If you just can't do the experiment, you just can't do the experiment. But spending money and thinking you got an answer when you didn't have enough power to get that answer is worse. You'd rather they didn't spend the money at all than spend the money on an experiment doomed to fail. So I would argue that a lot of what we can attempt to do in science is improve the reproducibility of our experiments. If you are using a kind of experiment that has a huge amount of variability associated with it, then that's the same thing as making these distributions really, really wide, right? So even if there is a mean difference, if those distributions are really, really wide, your ability to tease them apart is really limited. So having an experiment that produces much lower variability can help you to achieve better power with the same number of replicates. Okay, so with that, we come to the end of our lesson number 5, and maybe we all feel a little less adequate because every time I go through this experiment, I'm reminded of just how frustrating a lot of the tools we use in biomedicine are. A lot of the experiments we do are underpowered, and even if a difference exists, we might miss it. And in some cases, we might claim a difference even when none exists. We have to be careful on both fronts.
But how do we really, for us, we know we don't know how to interpret the result, though we have a nice result, but we don't really know if it's significant or not. So what do we do at that time?
Okay, well, yeah, sure. But you may have an experiment that has produced viable data, and if you just go running after every experiment with a t-test, then you're in a problematic space because lots of statistical models are available for evaluating any data set, and it might be that t-test is just not the right one. In this case, we're dealing with an ideal case for t-test, that the residuals in the model that we come up with are normally distributed. But there are lots of cases where people use t-test and they can't guarantee anything of the sort. So talking with a biostatistician is a good idea, but do it at the design stage. Because if you only talk to the statistician at the end of the day, after you've generated the data, and the statistician may only be able to tell you what your experiment died of. One of the better quotes from the field of biostat.
Okay, we know that the number of replicates is 5.
Yeah.
What if we don't know the effects that maybe the
Yeah, yeah.
Maybe the difference, people just say, we don't know the difference, they just use it.
Yeah.
Are you able to get the same?
Okay, so quite frequently, people are doing experimental designs and they don't know what the effect size is. They don't know if it's a mean difference of 1 standard deviation or a mean difference of 2 or 3 or whatever. A lot of people just use kind of a back of the envelope strategy for estimating the effect size in an experiment, and it's pretty weak. Generally speaking, I would say you can benefit from using prior data that have been collected. Like if you're doing your first proteomics experiment, you could download data that other people have submitted in a similar context and ask how much variability was there. When they saw protein differences, how big were they? So that is possible. Doing pilot experiments is kind of the next thing. If you know that you need to do a big experiment, but you're not sure how big an effect to anticipate, doing pilot experiments can help you to pin that down a bit. But all of these things help us to be more nuanced users of biostatistical methods. I hope that now that you know R, you can try to work a few examples of how likely am I to detect this given data that look like this or that. It can help you to say, well, look, we really don't know, so we better plan for a lot of replicates. Or instead of doing RNA-seq overall, we might focus on just a relatively small number of genes that are most likely to be involved. That kind of hypothesis direction to an experiment can greatly help the power that you can achieve with it, because multiple testing correction takes a much lesser bite out of your data. Okay, thank you, everybody.